acute management options to potentials for neurorestorative therapy. It's a great pleasure now to introduce our next speaker, Dr. Jeffrey Kocsis from Yale University, who's going to talk to us about the olfactory and sheathing glia as a therapeutic tool in transverse myelitis patients. Thanks.
Okay, so get the toys together first. Great. Okay, well, I too am honored and pleased to be here and have been enjoying the meeting, as I'm sure you all have. But I do want to say something. As I've been watching this meeting, I think as patients, of which there are many here, I want you to switch hats now from playing doctor to playing scientist, okay? And I say that because what I'm going to show you, and in the next talk there will also be some very basic science, and there will be some very clinically relevant work in the Dr. McDonald's talk, and I hope in mine as well. But I really want to go through with all the, I don't want to use the word hype, but all the publicity that's been going on with stem cells and with other types of cells for transplantation. I want to give you kind of an overview, focusing on work in my laboratory, about where we stand with transplantation in experimental models. I'll allude to a few clinical phase 1 studies that are going on, and John McDonald will talk more about that. But I just really want to give you a sense of what we might be able to expect in these model systems in animals, and where we can and where we can't extrapolate in terms of human disease like spinal cord injury, transverse myelitis, and multiple sclerosis. So for the first slide, which I'm controlling, I'd like to put this slide up. It's just a little reminder that we have an axon here, and this is from the central nervous system. It's actually the optic nerve, but it will look very similar anywhere within the CNS. And what you're going to see as we move along is that there are different types of cells that can form myelin. But normally, in the peripheral nervous system, as has been talked about, the Schwann cell is the key player. Dr. Griffith is a real maven in terms of Schwann cell, if he's still here. But in the central nervous system, it's the oligodendrocyte. I didn't put a picture in, but a very major difference is that the Schwann cell, an individual cell will make a segment of myelin. And I'll show you a segment of myelin in schematic form in a bit. An oligodendrocyte will make multiple segments of myelin. They have different proteins on their membranes and therefore different susceptibilities, particularly for immunological problems. And the olfactory and sheathing cell, I almost wish it wasn't in my title because we'll talk about other cells as well, but the olfactory and sheathing cell is very much like a Schwann cell under certain conditions. There's been a tremendous amount of excitement about OECs over the past couple years by the experimentalists, because normally the olfactory and sheathing cell does not form myelin. There's cells that are in our olfactory systems, both in the periphery, in our olfactory nerves, and in the olfactory bulbs, which are part of our brains. And olfactory nerve fibers they're very fine caliber, about 1/10 of a micron, tiny little axons that will enter our brain, transmit impulses for smell, form synapses. But the very interesting thing about the olfactory and sheathing cell and the olfactory system in general is that the neurons that are in our olfactory mucosa, they undergo mitosis, even in us. They degenerate, they regenerate. And the axons normally just grow back through the olfactory nerves. They go through the cribriform plate in the skull. They enter the central nervous system, and they make new synapses. So here we have this wonderful model of plasticity that's occurring in all of us. But the part that's interesting in terms of long-track regeneration, where a major problem is with spinal cord, any type of spinal cord injury, be it from MS or transverse myelitis, be it from contusive injury, is to get these long tracks back in place so that we can get information from up here, for movement, for example, down to appropriate levels on neurons that are remaining in order to make movements or to have appropriate sensation. But what the olfactory and sheathing cell does, and much of this work started with Ramon Cueto, there's work at the Miami Project, Jeff Raceman's group in London actually. What the olfactory and sheathing cells will do is they'll kind of enter and guide these axons in. If you cut a dorsal root in the spinal cord, the sensory fibers don't grow back in under normal conditions. But we have this natural process where there's something different about the OEC that makes it very conducive to elicit regeneration. And I'll show you some work that we've done with that and refer to others in terms of regeneration. But first I want to talk about demyelination, what cells can do to put myelin back on in experimental models. So if we go to the next slide, this is a little schematic that's evolved through the years, getting color on it. But at any rate, here's a typical myelinated nerve fiber. And I'm not showing you actual fibers. I left that slide out. But this segment of myelin, which is in black, it'll define two regions laterally called the node of Ranvier. It's a fundamental critical area for axon function. The node of Ranvier is where the action potential or the nerve impulse is elicited and initiated. And it's a very excessively specialized membrane. Sodium channels, which are the little batteries that will generate the nerve impulse, they're concentrated at an extremely high density at these red zones. It's about 1,000 to 2,000 voltage-sensitive sodium channels per square micron. It's a very high density of functional protein. If you go under the myelin, which is wrapping around and forming this insulating-like material, there are very few sodium channels here. So even if we take the myelin off, this membrane is called non-electrogenic. It can't generate an action potential because there aren't enough sodium channels. Fortunately, when you pull myelin off, sodium channels will either move laterally or they'll be synthesized and reincorporated. The density will increase, and you can get slowed conduction that has a little problem moving through, but you can get impulses to eventually cross this membrane. What normally happens, though, is we have a big generator of current right here with all these sodium channels. Sodium ions, positively charged, rush in. The flow of current goes to the next node of Ranvier, and the density is very high and very rapidly. 20 microseconds in a fast-conducting fiber, one can generate another action potential. This length can be as long as a millimeter. So if you do the calculation, I mean, you're moving across a football field in about a second because of myelin. Without myelin, the impulses would literally just be crawling along if they could conduct. In real time, a non-myelinated fiber, here's a meter, will go 1,001. They're moving like that. They're very slow. So what we get out of putting myelin in terms, it's happened through evolution, is that we can have a very compact, small unit, the axon, and because of this myelin, we can have very rapid impulse conduction. If we lose the myelin, the size of the axon would have to get larger, and we just would not be able to accommodate with the space requirements to have the cognitive and the motor functions that we have. When myelin comes off, there are two kind of hallmark signs that one sees in terms of functional or electrophysiologic, I should say, deficit. And they can be explained, I think, pretty easily with this diagram. That now what happens, we have our sodium channels here, we have current that's moving, but it gets distributed along this longer length. So it's distributed longitudinally. The same amount of current we have here is going to leave this membrane, but the density will be less. And so now what would happen is, if the density is so low that we just can't activate the voltage-gated sodium channels, we're going to have impulse failure. Hallmark, electrophysiologic hallmark sign 1. If we have a delay because now we're charging this membrane more slowly, maybe instead of that very rapid 10, 20 microseconds, it's 40, 50, 60. Then we're going to have conduction slowing. And if you accumulate slowings and blocks, you're going to have really completely disrupted the ability of this axon to send information from point A to point B, and that's critical. We do all the fine-tuning with our excitatory responses on nerve cells, and when the nerve cell body says to the axon, I want to go from point A 2, 3, 4, 5 feet, depending on how tall you are, down your spinal cord to get to those motor neurons, it wants to have that precisely timed. And it's disruption of the timing, not just the failure of the impulses, that can lead to severe neurological dysfunction. Now, I'm going to talk briefly here about one of the model systems we use. We use many. We contuse the spinal cord. We inject chemicals to demyelinate. We use genetic mutant animals, but I'm going to focus today on one. And the reason is that this is a very clean model to induce demyelination. It has some problems with it because we not only kill myelin, but we also kill other cells like astrocytes. But we use it as kind of a base to look at the extent of myelin that we can achieve with various types of cells, and then we go back and look in other model systems. So what we do is we routinely focally exirradiate the spinal cord, in this case of a rat. I'll show you a little monkey work later. And then we inject an agent called ethidium bromide, and it just gloms up the nucleic acids and kills the cells within a long white matter tract called the dorsal funiculus, in this case in the rat spinal cord. And then pretty routinely we stick to 3 days. We then go back in and we inject the various cell types that we're interested in. The model system was developed many years ago, a couple decades now, by Bill Blakemore in Cambridge, and it's really very good. You can see this dorsal region here in the spinal cord. This blue, this is a 1 micron plastic embedded section. Here's where all the fibers are, mostly the ascending fibers in this region for sensation. The descending fibers for the rat, the cortical spinal tracts, are located here. At higher power, you can see these rings, these blue rings of myelin. In our lesion model, we get areas like this, and they're long. They're 8 millimeters in length, kind of cigar-shaped, and there's virtually no myelin within this region. I know it's a little hard in this room. This projection is kind of small, so you have to take my word a little bit. But this lighter area, if you look carefully, there are little rings of completely demyelinated axons within the entire lesion zone. The reason why we exirradiate is that in rodent models, remyelination, spontaneous remyelination is very common, and it's partial. So if you really want to go in and assess the cells that you're putting in, you want to have a model system where, although you have to mark the cells, which is very difficult with reporter genes and other marking techniques, you really want to know that the cells that you put in are the cells that are myelinating. And we can see phagocytic cells here. They're chewing up the myelin. But we have these very large fields of no myelin. Now, in this experiment, what we did was we went in the center of that lesion, 8 millimeter lesion, so went into the middle, and we injected acutely isolated Schwann cells, which normally aren't associated with myelinating the central nervous system. But they do. Even in man, there's an occasional area where you'll see Schwann cells that have put myelin on. Part of the reason for that, although we have worked with cells that form oligodendrocytes, it's much more difficult to harvest oligodendrocyte precursor cells. And in diseases like MS, this thinking is simple for the immunologist here, but the antigens that are presumably responsible for some of the immunological problems are on oligodendrocytes and presumably not on Schwann cells. One does not get peripheral demyelination in MS. And when we inject these cells and wait 3 weeks, a single bolus at a point, not a huge number of cells, 10,000 cells, 3 times 30,000 cells. Then if you look at this higher power, and I'll show you electron microscopy, you can see these rings that have come back, the myelin, and when you look here in this EM, here's normal, here's our demyelinated, here's an electron micrograph showing classic Schwann cell-like myelination. One cell forming myelin, one myelin segment. You can see the axon, the thick myelin, the large nucleus, the cytoplasm. For the aficionados, there's a basement membrane, which is not normally formed in the central nervous system. And interestingly, there's collagen deposition, which you see in nerve, but you don't typically see in the central nervous system. If we then look, we put in a reporter gene, so we can see these beta-galactosidase products in here, knowing again that it's the donor cells that were responsible for the myelination. And I always pause here and make a point, because it may be going through your minds. This remyelination, which is extensive, it goes from the site of injection of the center of the lesion to all the boundaries of this 8 millimeter long lesion. It stops at the edge of the normal tissue. Absolutely critical if there is going to be consideration for a study to be done in man, of which there is, and there are, I should say. Because if these cells went in, continued to grow after they myelinate or before they myelinate, invade normal tissue, this could be very problematic and it could be destructive. But we, nor anyone that has used Schwann cells, we've had international conferences and we've all come to agreement upon this, see these cells stopping at the margin of the lesion, which is very good news. But the point I want to make that is less good, but I think important, and we should mention it, you can really just see macroscopically here the difference in these two profiles, or these two electron micrographs. Here the oligodendrocytes, you can see the axons are very densely packed, and they're not as densely packed here with the Schwann cells. Part of that may simply be because the nucleus and the cell bodies are so large, they push the tissue out. We don't see increases in volume, but there may be some axons that are lost. In the peripheral nervous system, we have lots of space. We can have a sciatic nerve, for example, that's very large, and it can be accommodated in our relatively large bodies. So the specialization that has probably come about through selection to have densely packed axons within the central nervous system is good for our cognitive functions and for our ability to move elegantly, but it's bad because if there are assaults on one oligodendrocyte, many axons are going to be disrupted. But the point here is that even if a percentage of axons that are in a chronic, large demyelinated lesion could be repaired by the Schwann cells, this may be good news. Several studies in animal models suggest that there is kind of a critical threshold of 10%. I can't stick to that. I'm telling you what they say in the literature, that if you lesion a long track in an animal model, let's say a rat, that you'll, of course, get functional deficit as you destroy these axons. But once you get below 10%, you have tremendous loss of function. So part of the rationale in the transplant community in terms of myelination is that even if we could get 20, 30% of demyelinated axons remyelinated, this could lead to potentially significant improvement in function and significant improvement in quality of life. Now, I'm primarily a physiologist from training, although we're shifting with time, and my interest in getting into this world of cell transplantation was initially primarily the physiology. If you put the myelin back on, and if they don't work properly, well, what good does that do? You can test the animals behaviorally, but I wanted to put microelectrodes in and see how they function. And our main mandate has been and will continue to be, with different cell types, not just to look for improved conduction velocity, but we also look very, very carefully to see if there are abnormal firing properties that are elicited from the myelination. I mentioned earlier that the sodium channel, there are many channel types, sodium channels, potassium, different subunits. My colleague and Department Chairman Steve Waxman has been cloning a zillion types of sodium channels. If you look at patients with neuropathic pain, the organization, the structure, and ultimately the function of not only the axon, but the whole system will change from rearrangements of the sodium channel and the potassium channel. So our question has been, do we see changes after remyelination? If the channels don't organize properly, they're going to fire abnormally. And the bottom line, and I'm not going to belabor this, is we've never seen that. When you look at these cells that have been remyelinated, first of all, this is a microelectrode that's recording field potentials. This is the normal dorsal columns. If you look at the little spacing here, it moves out. Each millimeter we step out with our electrode. It moves out at a very regular increment. We can calculate the conduction velocity about 15 meters per second. In the demyelinated fibers, these traces, the time base here is shifted, so they've been crunched down. It doesn't look as dramatic as it actually is, to about half. And when we move 1 millimeter, we have a huge jump, and then the impulses can't propagate all the way through the lesion. So we have conduction slowing and conduction blocked. And we are very pleased to see that after we put in the Schwann cells, we transplant them in with a micropipette, wait a few weeks. You can see that the spacing here in the remyelination group is very similar to that in the control. Statistically, it's been quantitated. You get pretty remarkable improvement in the conduction velocity. The impulses can now propagate a greater distance into the lesion. And then when we stimulate with various frequencies of stimulation, I didn't put that slide in here, but the fibers can follow high-frequency stimulation much closer to what the normal condition is. And that's critical, because getting one impulse through may be good, but most of our integrative activity is with small groups of high-frequency responses. And so the transplants are able to indeed follow high-frequency stimulation. Importantly, these are intra-axonal recordings where we get a fine electrode and we impale individual axons within the spinal cord. And here's a normal profile stimulating at 2 points. We know the distances we've carefully measured them. You can see here's where the shock is given, very short latency responses. In the demyelinated segment, propagating here, look at the very long latencies that we see. Just classic for a demyelinated fiber. If we look after the transplantation, the normal domain of the axon is conducting properly, and now we can get very rapid conduction. And I have to remind you that in our lesion model, there are no normal axons within that lesion zone. And if you pull the data together, it's very significant. One can really see that conduction, even on a single axon level, is dramatically improved. But not shown here, but implied, with these microelectrodes, if the cells begin to fire spontaneously, we'd see them, we'd hear them tick, and they don't. So there's no gross disorganization of the node of Ranvier. It appears to recapitulate a high density of sodium channels appropriate for rapid and secure conduction. Now, our interest is to ultimately, either by ourselves or encouraging others, to be able to find an appropriate model system to do phase 1 studies in terms of Schwann cells. We do have a study that's approved that Tim Vollmer, who unfortunately can't be here, was going to go over with you to tell you the basic plan. But something that I've done in my lab, this came out fairly well, about a year ago actually, in Journal of Neuroscience. What we decided to do is if we're going to try to do Schwann cells in a limited number of patients, and we're going to be taking these from most likely 40, 50, 60-year-old people, because the study will be autologous. We'll remove a piece of sural nerve, dissociate the Schwann cells, and then implant them into a MS lesion plaque that's well-defined in the forebrain near the anterior limb of the lateral ventricle. But the first question was, can they even work? I mean, we're dealing with rats, and they're relatively young. And so we set up a program where we would receive amputated limbs. That the limbs were obviously coming off regardless of whether we did this study or not, and from patients with diabetes and patients with vascular disease. And we then took out the sural nerves and worked out dissociation procedures to keep the cells alive. We then worked out cryopreservation, where we could store them and bank them. We then reconstituted the cells, and we were very pleased to see, this is chopped a bit, but here's our lesion again. Now this is in the rat, and the rat has been immunosuppressed with cyclosporin. And if you look in this little box, you can see the profiles here with numerous axons that have remyelinated with the Schwann cell pattern of myelination. Not the greatest marking technique, but we did an anti-human nuclear stain, and within the zone of transplant, it lights up. And we're pleased to see that if we looked with, in this case, field potential recordings, microelectrodes to look at small groups of axons, that here we have fast conduction, slow conduction, and demyelinated. In one group, we had pretty dramatic, not as good as rat, but improvement in conduction through the lesion site where the transplant was. And in another group, we had a fast component and a slower component. And the histology with the human cells that we transplanted show both myelination in some areas that were non-myelinated. It's our feeling that it's going to take a longer time for these primate cells to do their job, and we're in the middle of investigating that. But the good news is that there's clear evidence that the cells can survive for at least a couple months. We have to look longer. And that when they do survive, they're able to form myelin and improve conduction. I won't belabor this. This is just showing with single axon recordings, much like I did for the rat. Now, I put this paper from Nature Biotechnology up because a co-author is in the audience, Dr. Bill Fodor, and I want to give him some credit. But at any rate, in addition to looking at Schwann cells to remyelinate, we've also looked at olfactory and sheathing cells, as I've mentioned, and they remyelinate very similarly as the Schwann cells do. But there's another lesion model that I think is very important. And in addition to wanting to remyelinate axons, we certainly want to encourage axon regeneration. Olfactory and sheathing cells and Schwann cells have been known for some time now, if you transplant them in areas of transected spinal cord, that they can encourage axons to grow considerable distances, centimeters in the rat, which is a very significant proportion of the length of a rat's spinal cord. And one approach that we took was to utilize some cells that were developed in Dr. Fodor's group at Alexion Pharmaceutical. They're a transgenic pig colony, and in this case, the entire animal expresses a human complement inhibitor, the HCD59 gene. Their goal, and he's here, everyone can talk to him about the immunology of it, is to ultimately, with this approach and others, develop a pig colony that would be more conducive for xenotransplantation of cells or organs and possibly allow a vast source of cells to be produced. From my angle, I was interested initially just to see if these cells that have been genetically manipulated and engineered for the immunologic reasons I told you about, whether we could put them to work and actually achieve myelination or regeneration. So what we did was to purify, in this case, olfactory and sheathing cells, and in parallel we've done Schwann cells. We then transplant the cells in a site where we've completely mechanically transected with fine iridectomy scissors. We look before, after histologically. It's a complete transection of the dorsal funiculus. We then inject cells, boluses of cells, just lateral to the site, I should say rostral and caudal to the site of the transection. We then wait 5 weeks. In this case, we do have immunosuppression, and then we study both the anatomy and the electrophysiology. Anatomically, we're pleased that one can actually trace axons crossing the lesion, but as a physiologist, I really couldn't believe this stuff. I mean, you read things and you believe it, but if you don't do it, you don't really know. And so our game was to see if we could record impulses directly across that transection site. So if you do that, here's the spinal cord, it's been transected. If you inject control medium, so there's no Schwann cells or olfactory and sheathing cells, and you record, in this case, 5 weeks later, you get a shock artifact and a flat line, virtually all of the animals. But we're very pleased to see that a very significant proportion after we transplanted, in this case, the transgenic pig olfactory and sheathing cells, one could record impulse activity. Here we go 5 millimeters, but up to about 1.5 centimeters, the length of our chamber, across that lesion site. If you look anatomically and you actually count, very laborious work, Karen Langford in the lab has been doing that, one goes from about, just to give you some perspective, with this model system, and I think people are in agreement with transplant Schwann cells and OECs, you have a baseline level of maybe 60, 80 axons within that field, and you go up to maybe 1,000 or 1,200 if you're lucky. So enough to record impulse activity, but you're not, at this point, having thousands of axons that are regenerating, but it's a sufficient number of axons that others, including John McDonald, have shown functional recovery in very carefully controlled behavioral studies in the rodents that have had this procedure. Another point I like to emphasize, and I talked about it earlier, and I'll come back to it, we are not attempting to completely, at this point in time, recapitulate what was there. And in addition to seeing anatomical differences after transplantation of cells, that physiologically we were pleased to see that both with the transgenic pigs and with allogeneic grafts of rat cells, that the conduction velocity of the axons that have regenerated, which incidentally I didn't put the EM in, are remyelinated by the transplanted cells, the peripheral myelin-forming cells, that the conduction velocity is actually significantly faster than the control. So you say, well, what good does that do? You've got this fast conducting pathway, you've got a few axons. Remember, there is behavioral improvement that John will talk about, but my take on it is, and it's made me a true believer in rehabilitation medicine, because if we have nothing there, we have really nothing to work with. But if we can put in, albeit a limited pathway, rapidly conducting, there's so much endogenous plasticity in the central nervous system that we may be able to challenge that system with appropriate rehabilitative paradigms to improve function with this residual, or I should say this new circuitry that we've put in. And I really think that in this world of transplantation, with the limited technologies that we have, that as it is introduced experimentally into the clinic, that rehabilitation scientists are going to be very fundamental in testing efficacy in terms of these kinds of studies. Now I'm going to move on here kind of quickly. We are running a little late. We have begun a limited number of studies in monkeys. And primate work is, I believe, important, but it's not going to be the only answer. And it's very difficult and it's very expensive. And I think everyone in this room respects primates and rodents. And we really try to limit the number of animals we do. And I just want to reassure those, since this is not predominantly a scientific group, but an educated lay group, that our monkeys, and I think virtually all monkeys that have appropriate approval, are treated so well and with such care that I feel comfortable doing these studies. What we did was to design a very limited lesion where we make a small demyelinating lesion and an ascending sensory tract. And I'm very pleased that after the surgery, with or without the transplant, the animals do very well. I mean, the next day they're up and about. It's kind of, it's a small, it's a laminectomy. It's equivalent to having some relatively minor back surgery. And what we can do then is make areas of limited demyelination, and then the cells that we find in the lab from the rodents that we think may have potential to be looked at with clinical studies somewhere in the future, we can then implant them and see if indeed in a rodent model, which is different, in a primate model, non-human primate, which is very different from the rodent, whether we're going to be able to get remyelination. And we're struggling with this. We have some success. This is our lesion without exirradiation, but with the ethidium bromide injection in a monkey. You can see the higher power here, the non-myelinated axons, or demyelinated, I should say. And in several animals, we have clear clusters. These were injected with olfactory sheathing cells from the transgenic pig cells that I told you about that have the human complement inhibitor on them. And we're very pleased to see that we do get some remyelination, and we're trying to refine these primate studies. I think ultimately they're going to be very important. Now, I want to shift gears. I know we're talking about olfactory and sheathing cells, and trust me, this will link with them. I have a group of colleagues that were actually trainees in my laboratory. They're neurosurgical residents who are all now much more senior, about 5 or 6 through the years, and I have collaborations at the Sapporo Medical College in the Department of Neurosurgery. So what we are able to achieve there is to obtain human tissue. We're also able to have some parallel experiments going on. I want to talk a little bit about those. In brain, this is a stained section of a human brain. Here's rostral, caudal, front, back. The caudate nucleus is here. These are the lateral ventricles. There's a little region here called the subventricular zone. And you'll hear about it, and I'm sure you've read about it in the press. And there are little resident cells that hang out in there that are stem-like cells, neural precursor cells. They're not true. A true stem cell, as you'll hear about next, comes from the embryo. But these cells will migrate to the olfactory bulbs, and they normally do that. It's called the rostral migratory stream. And then they will differentiate into interneurons. So those other cells that I told you about, actually the axons growing into the olfactory bulb that will make new synapses, they're going to team up with cells from this rostral migratory stream. But many people have shown, we certainly didn't start this, Dr. Rao, there's been a whole slew of very important biological studies done, have shown that if you take these cells from animals out, put them in appropriate culture condition, you can make neurospheres. These little cells will form as spheres that float in your culture. You can then take those cells, pull them out of the appropriate mitogens, let them differentiate in tissue culture, and they look like neurons. They can turn into astrocytes and the oligodendrocytes, very few oligodendrocytes, but some, and you can push them into a neuronal lineage or a glial lineage. So we basically repeated that. Dennis Steinler's group has done this in human tissue, even cadaver tissue. We got frontal lobes from patients that had tumors, and their frontal lobes are going to be removed nonetheless. And we then generated neural spheres. Here's one cell, and we clonally expanded them so we could take individual cells, let them grow up, so to speak, in clusters, dissociate those and inject them. And I thought for sure that we were going to see some neuronal differentiation, some oligodendrocyte differentiation, some astrocytes. But in that lesion model in the rat with immunosuppression, the predominant, not exclusive, but the predominant myelinating phenotype was this peripheral-like myelin-forming cell. We did have some oligo-like myelin, but the predominant cell from the adult human brain, cells that were taken out, put into this axon-enriched area in a rat, formed a lot of myelin, but it was from a neural crest, not a neural tube derivative. And there's been publications in the basic biological literature of talking about how plastic these cells are, even within the adult human brain. I should also mention that this is preliminary, but when we look at the shape of the nucleus, and there's a couple other features, it's really, first of all, it's hard to distinguish a Schwann cell in culture from an olfactory and sheathing cell. They have many of the markers we use, they're the same. But there are morphological features on these peripheral myelin-forming cells derived from adult human brain that look more like an olfactory and sheathing cell than they do a Schwann cell. But the point here is that if indeed peripheral myelin-forming cells, when we transect spinal cord, can encourage long-track regeneration, this may be a potential source of cells that we could harvest, expand, and be able to use in clinical studies. We did a series of experiments that's been published in abstract form. We still have a couple more controls that I think is important. We took another monkey type, the marmoset, a very small monkey. This was all done with my colleagues in Japan. I usually spend a month or so a year there. And these guys are great. They're terrific neurosurgeons. So they got their ultrasound probes out, went in, did a pretty accurate biopsy here in the anterior limb of the lateral ventricle from a marmoset. This is a little diagram showing where the lateral ventricle is from the ultrasound probe. Then we took those cells, expanded them with conventional techniques, developed neural spheres. The monkey was doing very well. The procedure for the biopsy is relatively innocuous. It could conceivably be done with some risk, but minimal risk in man. And when we injected those cells into the ethidium bromide lesion of the same animal, so this is autologous, we were pleased to see that we had fields of axons that had myelin on them. So I think that the possibility of doing such an autologous approach, I think, is there, and we're very pleased. In a small number, half dozen marmosets, we were successful in this regard. Here's an EM of some of that material. Now, I want to close. Time is pushing on here, even though I did get started 15 minutes late. We have some preliminary data, and I want to shift gears just a little bit. There is much excitement with bone marrow, and I will show you a little bit of this data. We just published a paper in Glia where we fractionated bone marrow sort of conventionally, the way other neuroscientists will do it, and we take out a so-called lymphocytic fraction of the mononuclear layer. And before we embark upon separating the individual cell types, and we may not, because there are companies out there now that do this and we can collaborate with them, but we were just curious what would happen. There's been much talk about stem cells and bone marrow. So what we did was to inject the bone marrow, first of all, directly into our lesion model, and we were very pleased to see that we established very nice myelin. This has been published with direct injection of it's not quite a raw bone marrow. It's a bone marrow that's been kind of cleaned up, so there are no erythrocytes, red blood cells, and platelets and junk in it. But it's a really pretty undefined layer. We really have to find out what's in there. But the literature suggests that there is a stem cell in there, and there may be neural-like stem cells within there. But we were very impressed that we were able to get relatively extensive remyelination. We have an abstract we sent in and some preliminary work that we're finishing off in a paper that's out for review. We decided to inject this bone marrow intravenously to see what would happen. There have been a couple reports that with IV injection of the same fraction from bone marrow, one can get neuron differentiation within brain. And we're very excited because with that lesion model, now the blood-brain barrier is probably a bit compromised in this with the exirradiation and the ethidium bromide, but if you look at the spinal cord, this is following an intravenous injection of a crude bone marrow fraction, partially separated. You can see the little blue spot. This is beta-galactosidase. We had these cells had a reporter gene inserted into them. When you react it turns blue. If you look here in these thin sections, you can see the little dots strongly suggesting that indeed the cells that we put in were the cells that formed the myelin. There's some improvement in conduction, but actually, let me go back. This is out of sequence. But both Schwann cell-like myelination and oligodendrocyte-like myelination, we haven't done the immunostaining yet, are scattered throughout the lesion. So the prospect, I'm not saying it's going to happen, but the prospect of being able to get stem cells even autologously from bone marrow and to treat them properly and to infuse them intravenously to home to the site of the lesion is the kind of thing that scientists are excited with playing with in a laboratory. To extrapolate that to a clinical situation, there's going to be an awful lot of thought and work that's going to need to be done. So I'm going to finish off here, and I just want to let you know that not only my group, but several groups, and you'll see the exciting work that John is doing, are really focusing, looking at the possibilities, not just for the good things that cells might do with transplantation, but we're being a little guarded, and we want to look for some potential negative things as well. Thank you very much.